the recording. So could you introduce yourself to our listeners?
Yeah, hello, good evening. I'm Kerstin Göpfrich, and I am a group leader at the Max Planck Institute for Medical Research in Heidelberg. And my group is basically working on the bottom-up assembly of synthetic cells. And for this, we use different kinds of tools, but actually our favorite tool is DNA nanotechnology, which we use to build functional components for synthetic cells.
So that's sort of why we were super interested to have you on our podcast, because your research group is in creating these sort of synthetic cells, which are sort of like, this is so cool, like very large, using sort of laser printing, microfluidics, and synthetic biology. So DNA fluidics or synthetic biology. So could you tell us a bit more? What is a synthetic cell, and why are you working on synthetic cells?
Yeah, I guess what is a synthetic cell depends a little bit on whom you ask. So basically, if you ask me, I would say it's kind of a model system of a living cell, which is assembled from the bottom up. So basically from molecular components, which you start to put together like the pieces of a puzzle to make something which has some functions at least of a living cell, but ultimately also all functions of a living cell. So ultimately it should deserve the name synthetic cell in its true meaning, so to say. So basically we start with a lipid vesicle and equip it with more and more lifelike functionality, if you wish. And so in the way I understand it, the synthetic cell is really an entity that's assembled from the bottom up, which we can use as a model system of a living cell with reduced complexity, and therefore maybe understand some of the biological processes, but also really engineer something entirely new. And I mean, then of course you can ask yourself, okay, when are we there? When do we actually have something which deserves the name synthetic cell, let's say, because synthetic cell is quite a broad term. And this, of course, comes down to the definition of life in itself, right? When would you start calling something a synthetic cell? Well, at the point when it's alive, I would say. So so what do we need for that? I mean, there are, of course, lots of different definitions for living systems out there, and for me the most the best working definition, let's say, is a self-replicating entity capable of evolution. And so this is really what I think we should be after when we construct these synthetic cells from the bottom up. But just to give a broader picture, I mean, obviously a synthetic cell can also mean that you start with a living cell, just a biological cell as we know it, and then you use tools like genetic engineering to really modify it and make it considerably different from life as we know it, and really assemble a synthetic cell from the top down, so to say. So that's kind of the other side compared to this bottom-up approach that we are pursuing.
And your group uses a lot of laser printing, right? Could you explain a bit more on the role of laser printing for synthetic cells?
Yeah, well, laser printing is actually kind of the latest addition to the toolbox that we've been using to actually build synthetic cells. And it actually was born out of the idea that you quickly realize that if you look at a cell, its interior is highly complex, but also highly structured and highly organized. And so we thought basically, okay, what can we do inside a synthetic cell? Well, obviously we can encapsulate components inside of a lipid vesicle or a cell-sized compartment, but actually once you've encapsulated them, it's quite hard to position them in space and time in this because of the physical barrier of the compartment, so to say, right? And so we thought, okay, maybe a way around this is actually to use light, because light has obviously access to the interior of the compartment. So to use light and laser printing more specifically to really arrange components in space and time at the interior of a compartment. So we were asking ourselves, can we use direct laser printing, so 2-photon direct laser printing, to actually assemble structures on the inside of a lipid vesicle, really by 2-photon polymerization. So by using light to trigger the polymerization of a substance in 3D space. And turns out that this actually works. So first of all, we started off with making some simple proof of principle structures using like a common photoresist composed of PECDA and the photoinitiator lab, which polymerizes into a hydrogel structure, so to say. We made like smileys and letters and cubes and, you know, like 2D and 3D structures. But we also started to make like one of the first, or to print an early, let's say, functional architecture, namely a transmembrane pore, where we used 2-photon laser printing to kind of print a nanoporous cylinder across the lipid membrane. Turns out the vesicle is actually stable during this process, which surprised me quite a bit at the beginning. But yeah, cool that it works.
So yeah, so when you're you mentioned that laser printing is sort of the more latest addition to sort of the toolbox. What was the initial toolbox made from? What were sort of the more long-standing sort of components of the toolbox?
Well, to take on kind of a historical perspective, or to look at it into the past of what, or the past and present of bottom-up synthetic biology, quite commonly, a quite common strategy is to actually start with components that are isolated from cells. So basically proteins, those are the functional machines inside cells. So it's kind of makes sense to take those and actually reconstitute them or put them inside of lipid vesicles, and then basically study their function and their behavior in kind of a minimalistic environment where you know exactly the composition, the number of proteins, their concentrations, the types of proteins that you put in, and you kind of strip down complexity to find a minimal set of proteins which can achieve a certain type of functionality. And this led to actually a lot of biological insights also on what these minimal systems could be. So for me, I was always fascinated by the idea to use completely different kinds of components to build up the same functionality, to kind of have an orthogonal system, which is not based on the components that nature already uses. Because if you want to make a synthetic cell in its true meaning of the word, right, truly synthetic, if you want to make a truly synthetic cell, you should really also make your own components, right? And so kind of driven by this vision or this idea that you know, a synthetic cell should be truly synthetic, we also I started off to make our own components, mainly from DNA nanotechnology. So for me personally, this was the first tool, like to actually start using DNA not just as an information storage material, but actually as a functional material, which you can like use to make first of all 3D shapes, like with DNA origami, for instance, and then you can impart function on these structures. So this was really the earliest tool.
Yeah. Can you explain more about the DNA origami to us?
Yeah, so DNA origami is about repurposing DNA as a construction material. So taking it out of its context in genetics and information storage, and really using it for molecular architecture, so to say. So basically, based on the complementary base pairing of DNA, so just the fact that A binds C and G binds C, you can start to make architecture. So to give you a very simple example, of course, we know if two strands of DNA are complementary, they will start to bind. And now imagine you have got two strands which are complementary, and now you extend the ends of these strands with a random sequence that is not complementary to one another. Then you are creating kind of a binding site for a third strand of DNA, which is complementary to both of these overhangs, so to say. And then you already create something like a 3-armed nanostar. And this is like a nucleic acid junction, so to say. And this was actually the idea that Ned Seaman, who sadly passed away earlier this year, came up with. And this is, I would say, the revolution, you know, the revolutionizing idea to use DNA for architecture. And then with DNA origami is a slight modification of this, which comes from Paul Rothman, that starts with actually a long single strand of DNA taken from a virus typically, and it's about 7,000 bases long, and you take about 200 short synthetic oligonucleotides, which you use to kind of fold this long piece up. And because of that is also where the name origami comes from, right? You fold this long piece of DNA with many, many short strands that are complementary in defined positions.
I'm curious about sort of how this sort of essentially like three-pronged, like you called it, I believe, a nano star, was it? How is this sort of beneficial towards creating like structures with DNA in comparison to, let's say, this like long viral strand that you can then fold?
So actually, I can give you an example from our own work. So we were very recently actually working with this three-armed star, nanostar. I don't know if it's the right term, but three-armed branch structure of DNA. And we used it actually to polymerize it into a hydrogel. So imagine you've got this three-armed nanostar, and then you make the sequence overhangs kind of self-complementary, so that this nanostar can polymerize into a huge mesh. Then it just polymerizes, and you can actually create a DNA-based hydrogel. So you can actually make a physical hydrogel, which you can punch with a pipette tip or so. And now if you introduce, for instance, photocleavable groups at these arms of the nanostars, then you can actually use this to print. And then we are back to the printing story. Basically, you can use laser light, the light of a laser, to actually erase predefined structures from this hydrogel. And then you can do this on the inside of a lipid vesicle, for instance. And the advantage of using DNA compared to those more commonly used photoresists is that, first of all, it doesn't require free radical polymerization, which is beneficial in the biological context. It's not so damaging. And secondly, it's a natural material, so that's great anyway. But third, you can actually quite easily integrate dynamics and functionality because of all the stimuli responsive motifs that are out there for DNA right now. So I think DNA is quite a cool photoresist, and I'm sure we'll be using it more in the future. And really, this can be based on like a very simple motif like this three-armed nanostar, which you can already do quite complex things with it.
Can you imagine for that to be sort of, would you say that there's likely to be an increase or more of these motifs coming up? Or do you think that the work will be driven more by the motifs that are already sort of in place or are being experimented with?
Well, I'm sure like there will always be modifications of the things that are out there, right? So basically, I mean, if you think about it, right, like just take the example of the nanostar again, right? In principle, you've got binding sites, you've got ends of DNA in three positions, right, at the ends of the three arms. So I mean, people could use it and are actually using it to simply link components of their choice, right? So you could say, because of the large toolbox for chemical functionalization for DNA, which is out there right now, you can actually say, okay, on arm number 1, I want a protein, I don't know, streptavidin. On arm number 2, I want, I don't know, a fluorophore. And on arm number 3, I want something else. And now this seems quite random, right? But if now think about, for instance, positioning or bringing into close proximity, like certain antigens, for instance, right? You could really control the spacing between these antigens and answer questions like, how does valency correlate with cell response or so? So you can really start addressing biological questions by simply using DNA as kind of a linker or kind of as a scaffold to position secondary components, right? And I think this is where the true power comes in, to actually just use it as a breadboard to position the components that you like at the distances that you like, distances and valencies.
And we're also wondering, what are the current challenges facing the production of synthetic cells? Because all this technology seems to be like very exciting and have a lot of possibilities. So we're just wondering what are the bit of the challenges in the production of synthetic cells.
Well, I'll give you my very personal perspective. So far, bottom-up synthetic biology, and I talk only about bottom-up now, has often taken a modular approach, right? For instance, you make a module which for energy generation, you make a module for migration or movement, you make a module for cell division, and so on. So basically, we've studied all these lifelike processes in isolation, which makes total sense if you want to understand certain functions. However, you also quickly realize when you start actually recombining these functions into multifunctional systems, you quickly realize actually that, well, a cell is more than the sum of its parts, right? And we are quickly actually reaching the horizon of known structural function relationships. And I believe that here we are really at the crossroads, and we should be actually thinking about new types of approaches. And in my opinion, the most promising one is actually to think about integrating a fundamental characteristic of life, namely evolution, into the design process of synthetic cells itself. So to really, because evolution can deal with unknown structure function relationships, and that is what makes it so powerful, right? And I mean, since we know what we are after, I don't think that we'll do a complete random walk, and this is why I also don't think it will take billions of years until we get further, right? We know what we are after, so we can actually use directed evolution tools, which of course speeds up things tremendously. And so, yeah, I think going away from this pure rational engineering approach and integrating evolution is I think really powerful here in this context. And yeah, as the biggest challenge is really to recombine multiple functions at the moment, especially if we stick to a modular approach.
So what type of functions has how far has your lab come in creating these sort of synthetic cells, and have you already sort of experimented with directed evolution approaches?
So I actually started off in the group of Ulrich Kaiser in my PhD, where we made functional nanopores from DNA nanotechnology, yeah, back then actually those were we used them as sensors, so we considered using them as sensors for instance nanopore sensing, and for me they were often components of synthetic cells, and I mean this is then the direction that I pursued, and in the meantime. We made other components, like for instance DNA-based filaments, which mimic like cytoskeletons of living cells, or also exoskeletons, or structures which determine the shape of the lipid vesicles, or enable the transport, the intracellular transport, so to say, of vesicular cargo along these filaments. So really, cytoskeleton mimics have been a bit of a focus of our work lately. But then, of course, also like DNA-based photoresist, which we can print on the inside of lipid vesicles, and yeah, so the types of functions that we realize are quite diverse. So going away from DNA nanotechnology, for instance, we also realized the division of lipid vesicles, which may seem crucial towards constructing a synthetic cell. So it's really based on physical principles that the mechanism that we realized, so not really protein-based machinery, but just exploiting membrane biophysics, so to say. So I would say there is a few functions that we can do, but there is definitely also a long way to go. And that's what's kind of making it also very exciting, and I would say a fruitful challenge for many years.
So where do you see sort of the I guess sort of the next steps after creating these cytoskeletons and DNA filaments? What do you see in the future of synthetic cells? Like obviously, like the initial, let's say, directed evolution approaches. What would you study first, and how would you approach it?
Well, I would say so. I mean, there is always a lot of push for actually applications of synthetic cells, right? So what could be the applications of a synthetic cell? Where could we be using it? How is it going to revolutionize, for instance, biomedicine or environmental challenges also, right? And this is all very exciting, but But actually, for me, I think what's much more powerful is the kinds of tools that we develop on the way, right? In the end, what we do is we develop DNA nanotechnology further, we develop certain microfluidic functions, we develop laser printing for applications, for instance, on the inside of compartments and so on. And these are tools that are directly applicable to either study biological cells, life as it is, or actually applicable as technology. So for instance, when we studied cell division, synthetic cell division, of course we did it because we were fascinated by bottom-up synthetic biology, but at the same time, we also found out that actually the mechanisms that we looked at are quite useful for as an osmolarity sensor. So, you know, like current osmometers require freezing of the sample, and we realized that we can use our synthetic cells, for instance, to kind of measure osmolarity in a way that doesn't require freezing of the sample, that is compatible with live cell microscopy. And you know, maybe like solutions like these can simply help us to address questions in biology or be used as technologies much before we arrive at the end goal, right? So rather than saying, you know, once we have a synthetic cell, we can do X, Y, Z, I think let's not wait. Let's use the technologies that we develop already now to solve real world challenges, right? And then once we, and rather do the synthetic cell for blue skies reasons, right? Because I don't know, like, I mean, depending on the application, you may not even want a self-replicating entity, right? If you think, for instance, about I don't know, certain applications in drug delivery, right? You want to deliver a drug and maybe in a smart way, right? Maybe you want a sophisticated release mechanism or so. You could call it encapsulated drug in the inside of a lipid vesicle and then program that vesicle kind of as a synthetic cell to deliver the cargo to where it's needed. That could be an application, but then you may not want this entity to self-replicate. So in the true sense of the word, in my opinion, that wouldn't really be a synthetic cell, right? So, well, maybe you can call it like that. I don't know, and I don't care so much. But I mean, still, it's about well, at least what's fascinating me is to really build a lifelike entity which is self-replicating and evolving. And you know, you can just let go and see what happens. Like this is, I think, really driving me.
Yeah, it sounds very exciting. And yeah, with the creating new synthetic cells, we're also wondering what sort of feedback have you your research has gotten back from the ethical perspective of creating like a cell from scratch, if there is any form of criticism or comments that you've like heard over the years. We're just interested to know it.
This is raising a really interesting point. I mean, so yes, I have, especially in discussions, you know, I have heard things like, "Are you playing God?" You know, like to that point. And I mean, it's I think a really important question to ask and an important point to discuss also with society. I mean. So right now, if we write grants, if I write a grant, if I do a grant application, there is no box that I can tick, right? Because I'm not really working with a living entity, right? And everything that's, you know, everything that we perceive as, you know, should be protected is typically dealing with living systems, right? If you work with mice, if you work with engineered cells, if you work with, of course, like humans or whatever, of course, you have to make an ethical assessment and go through the whole procedure. If you work with molecules like we do, there is nothing, right? I mean, we work with molecules in the most literal sense. And so I would say at this point, like there is simply not really an ethical concern because we literally work with molecules. Well, if we add a drop of dish soap, then everything we've created is dead, although it has never been living, so I can't even claim it's dead. So in that sense, at this point, I think we shouldn't over-regulate things, right? Because there's no need to. However, of course, once we reach a point where, you know, our synthetic cells fulfill a definition of life, a definition of life, then of course this completely changes, and we should have this in mind. And I would say that the type of research that we do is actually beneficial to understand, say, certain principles, for instance, of evolution. And I mean, this is really crucial to understand if we release any type of synthetic entity, any type of synthetic cell, for instance, in the environment. So again, we have kind of a model system. But still, of course, like once we say, okay, we want to get to a point where we can actually evolve greater complexity and maybe also see surprising things that we did not program into our synthetic cells. So once we really step away from a pure rational engineering approach and, just let go, then of course we need the corresponding regulations in place. So this is really an interesting discussion, which we are also having a lot. And actually in some of our consortiums, ethics groups are specifically included. And we do have these discussions at conferences, at joint meetings, and so on. But it's really great to be able to involve the public as well.
So that's really interesting. So you mentioned how the ethics sort of you already kind of involve the ethics committees within your sort of discussions and the research that you do. Can you tell us a bit more how that sort of takes place? Like what role sort of do scientists play? What role do the ethics committees play? How they are all sort of made up in order to decide how to regulate what you will eventually end up getting at?
Well, so I would say in the first instance, it's about communication, right? So basically, for instance, I can give you an example in a consortium that I have been part of, the Max and Bio network, which is kind of a synthetic biology network of the Max Planck Society. We've had just one group from Erlangen actually, who was an ethics group, right? And so then it was about neutral information, right? So they joined our meetings so that they had an idea of what was going on. At the same time, they kind of did studies on like the public perception of the ethics side of the whole research that we've been doing. So part of it is really about information sharing, and I think this is the most important point at the current stage of the project. So right now, as I said, I don't think it should be about regulating, right? But this continuous information, I think, is really crucial to move forward.
And we're just curious because read that you started with a bachelor's in physics and molecular medicine, and then you moved your way to a field of synthetic biology. And we're just curious on how did you find the field, or what was exciting for you that drove you to get into the synthetic biology?
Well, I would say I was always fascinated kind of by the really big questions, right? Like questions that, my parents couldn't answer or nobody could answer, right? Like questions like, what is life? Can we build a living cell? How did life emerge on Earth? Like all of these questions that, nobody has an answer to, but that are somehow inherent to humanity, we've been asking these questions since thousands of years probably, right? And it's really awesome to see how science now starts to have the tools to actually approach them. And so I actually started studying molecular medicine, and then I started studying physics, and it seems like none of these, the endeavor is simply really interdisciplinary, and it's cool to see that we can start to approach these questions now. And I think this fascination for the big questions is really what drove me into the field of synthetic biology.
I think those were all our questions so far. Yeah. It was a very nice discussion and really enjoyed it. Thank you for being on this podcast.